The overall goal of this procedure is to provide an easy and generalizable protocol for the preparation of monovalent quantum dots for single-molecule imaging of target membrane proteins in live cells. This is accomplished by first introducing a stoichiometric amount of a long polyphosphorothioate oligonucleotide ligand to a quantum dot. The long DNA completely wraps the quantum dot and prevents the binding of a second DNA molecule, thereby generating monovalent quantum dots exclusively and quantitatively. Separately, benzylguanine functionalized DNA bearing complementary sequences is prepared to target snap-tagged membrane proteins expressed on live cells. Next, the sequential treatment of live cells expressing snap-tagged membrane proteins with benzylguanine DNA followed by monovalent quantum dots leads to selective labeling of the targeted receptor. The final step is to image the target protein distribution and diffusion trajectories via single-molecule fluorescence microscopy. Ultimately, real-time dynamic spatiotemporal information of target proteins in live cells is extracted to reveal how the target molecules work in vivo.
The main advantage of this approach to synthesizing monovalent quantum dots is its simplicity and general accessibility. Unlike many other approaches that require special skills or equipment, this approach is relatively straightforward. As a consequence, it should be available to any lab and from any discipline.
We first had the idea for this method when Zeva and I began discussing how our expertise on DNA nanoparticle synthesis could be combined to better control the reactivity of nanoparticles. We carried out the initial proof of principle experiments together over a weekend. Encouragingly, our first trial worked successfully.
In this video, we use monovalent